Well, this is the second part of lecture of today, 20th of April, 2020. We will study about the factors that stimulate the acid secretion in the body, not on the cellular level. The first side is the cephalic influences of stimulate the gastric secretion, the gastric part, the intestinal, and the others. For example, hypoglycemia, alcohol, or caffeine. What we mean by the cephalic influences, the presence of food in the mouth, the smell, seeing the food, it will stimulate the gastric secretion and increase the motility, so by reflexly through the efferent fibers of the vagus. As I told you before, when you see the food or smell it or eat it in the mouth, this will stimulate acid secretion reflexly through the efferent fibers of vagus. The cephalic influence responsible for third to fifth of acid secretion responds to normal meal. Psychological state, for example, when the patient or the person, normal person, feel angry, this maybe increase the secretion. When he's in a state of fear or depression, maybe this decrease this part of stimulation. The gastric influence, when the food in the stomach, it will accelerate the increase in the gastric secretion through a local reflex within the wall of the stomach because of stretch of the wall of the stomach, and also because of presence of chemical material and food particles. All these will stimulate the gastric secretion by the parietal cell stimulation, and the product of digestion also increase the secretion of gastrin, augment the acid flow. Intestinal influences has the opposite action. It will inhibit the gastric secretion by a neural and hormonal mechanism, for example, by peptide YY hormones. Because when food reaches to the intestine, there is no role for the gastric acid and start the function of other enzymes. As you know from the anatomy, this is the stomach, and there is the duodenum, the first part of a small bowel, and there is a forming of C shape where it is the place of the pancreas. It is a glands which share its secretion in the digestion. And you see this duodenum, there is an opening which is called ampulla of Vata, controlled by sphincter, which is called sphincter of Oddi, and it is a site of secretion of pancreatic juice through the pancreatic duct, and also the secretion of bile through the common bile duct. Let's take a few notes about the pancreas. As you know, it is a big gland in the C shape of the duodenum, composed from the acinar cell, pancreatic duct of Wersings. There is two pancreatic ducts, the ventral and dorsal, and they are combined together to form single pancreas that open in the ampulla of Vata. And there is, of course, the accessory pancreatic duct. So the result is combination of ducts into one opening. So pancreatic duct with the common bile duct, it will open in the ampulla of Vata in the duodenum, and this papillae is surrounded by circular muscle, which is the sphincter of Oddi. If you do an endoscopy in the second part of the duodenum, you will see this structure, the round oval structure. This is the ampulla of Vata. Of course, the pancreas is a gland, has an endocrine portion and has an exocrine portion. The endocrine portion that secrete the insulin and the glucagon. The exocrine portion that secrete the enzymes, which is pulled in the duodenum, and this under the control of reflex, GIT hormones, secretin, for example, to stimulate the bicarb, cholecystokinin, which is stimulate the secretion of pancreatic enzyme. So pancreatic enzyme is act in an alkaline media. The pancreatic juice is an alkaline. The pH is 8 or more. The amount is about 1,500 ml per day. One of the enzymes is the trypsinogen. It is the inactive form. By the action of enteropeptidase change to trypsin, and the trypsin will activate the other inactive form, the chymotrypsinogen, to form the chymotrypsin. These two enzymes, it will act on the protein digestion. The other enzymes is the carboxypeptidase and co-lipase, lipase from its name, share with the pancreatic lipase for digestion of triglyceride. There is also pancreatic amylase, which is act on the starch. Why these enzymes not digest the pancreas? Because you know pancreas contain of protein and fat. Because there is a trypsin inhibitor, there is factors which is protect the pancreatic tissue from the auto-digestion by its own enzymes. So potential danger of the release into the pancreas of small amount of trypsin is apparent that could digest pancreas is suppressed by the trypsin inhibitor. So when there is a disease of pancreas, for example, pancreatitis, that means that there is increase in the level of circulatory digestive enzymes like amylase and lipase, and there is less trypsin inhibitor. The other gland which is present is the liver. Which is a big organ, has many functions: formation of and secretion of bile, nutrition and vitamin metabolism, and activation of various substances like drugs or toxin, has role in immunity because has type of cells called Kupffer cells, like the action of macrophage. This is the liver. This photo show you the what's called a biliary tree. As you see, there is a small canalicular which is compound to form the left and right hepatic duct, share to form the common hepatic duct, and this will get the cystic duct from the gallbladder to form the common bile duct, and the common bile duct would share with the pancreatic duct to be open in the ampulla of Vata in the medial wall of the duodenum. So the role of liver in digestion is by secretion of the bile for digestion and absorption of fat. There is a bile acid, there's bile pigment, the amount is about 500 ml per day. And we will see the journey of the bile. The GIT part of bile, it's reabsorbed again and returned to the liver, and this is what's called the intrahepatic circulation. You know, the bile is released first from the hemolyzed RBCs. There will be release of hemoglobin. The globin will be apart, and there is the heme. The heme composed from the iron, from the precursor of the bilirubin. Here, the bilirubin will be unconjugated bilirubin, or called indirect bilirubin, which is water, not soluble in water, so it cannot excrete in the kidney. Then it will be picked up by the liver, and by the presence of enzymes, which is called the glucuronidase transferase enzyme, is changed from unconjugated bilirubin to conjugated bilirubin, which is water soluble, and then it will be passed through this biliary tree that you saw in the previous slide and reach to the duodenum through the ampulla of Vata. In the duodenum, they form what's called a micelles. We will see in the second slide. This is structure contain the bile, contain phospholipid, contain the fat that release from the stomach, fatty acid, contain a fat-soluble vitamins. So it's arranged for structure which hold the bile and these components all to the terminal ileum where the absorption is occur. 95% of it will be absorbed. 5% only will continue its journey to the in the colon, and in the form of the stercobilinogen by the effect of colonic bacteria change to stercobilin, and it's responsible for giving the brown color of the stool. Part of it will excreted in the kidney in the form of urobilinogen to give the color of the urine in the form of urobilin. And as I mentioned in the previous slide, part is reabsorbed and picked up by the liver again, which is called intrahepatic circulation. So there is a bile salt, bile pigment, or the bile, which is alkaline, 500 ml. And now imagine yourselves in the duodenum. As I said, there when the bile is released from the ampulla of Vata, it will form, this is what's called the micelles or compound or structure, contain fatty acid, monoglyceride, bile salt, phospholipid, cholesterol, vitamins, fat-soluble vitamin. and all these will be go to the terminal ileum through the brush border, and it will be absorbed to the circulation at about 95% of the bile. 5% will continue its journey to change to the stercobilin. If there is any defect in the bile secretion, there will be fat malabsorption and fat-soluble vitamin malabsorption. So when this physiological process is disturbed, when there is defect in the metabolism of the bile, there will be a jaundice, and the jaundice is a sign or symptoms that the patient will be complain, and it is a yellowish of the skin, sclera, and the mucous membrane due to accumulation of the unconjugated bilirubin or the conjugated bilirubin. When the total plasma bilirubin more than 2 or more than milligram or more than 35 micromole, it will consider to be more than normal. The cause of jaundice depends on the defect in which steps that we mentioned. For example, excessive production, that's mean in case of hemolysis when there is more RBCs destructed. If it is more than 6 times of normal, the liver cannot pick up all the unconjugated bilirubin. Or sometimes there is a decreased uptake in the hepatic cells. The liver cannot take it all, or there is defect in the enzyme, so there will be no conjugation. In the first 3 points, there is increase of unconjugated bilirubin. While when there is defect in the secretion after conjugation, or there is intrahepatic or extrahepatic obstruction, this lead to increase of conjugated bilirubin. How there is regulation of biliary secretion? The fatty acid, amino acid will, when reach to the first part of the duodenum, they stimulate the secretion of cholecystokinin, and this will cause contraction of the gallbladder, and the gallbladder will produce this concentrated bile to the common bile duct and reach to the duodenum where they form the micelles. The bile production is increased and stimulated by vagus and secretin secretion. So what is the function of the gallbladder? The bile flow to the gallbladder when the patient is fasting and the sphincter of Oddi is closed between the meal. The function of gallbladder, it is a storage. It is a store of the bile and increase its concentration of the bile by water absorption. When the food enters to the mouth, there will be decreased resistance of sphincter of Oddi and release of the bile. Of course, the presence of fatty acid, amino acid in the duodenum will, as I mentioned, release the cholecystokinin, cause gallbladder contraction, and release of the concentrated bile to the duodenum. Here I will finish this second part of this lecture, and if you have any questions or problem, you can ask me.